Hepatitis B and C viruses chronically affect about 3.5% of the world population, causing severe liver pathogenesis and more than 1 million deaths every year. Better HBV vaccines and the development of an HCV vaccine are urgently needed to stop this life loss and eradicate these pathogens. Towards the SmartVac project, Dr. Nichita and her team explores plant and algae as cost-effective system for production of novel HBV and HCV antigens. This way, they hope to contribute towards the elimination of viral hepatitis as public health problem by 2030, an ambitious target set up by the World Health Organization in 2016. Keep watching Research in the Spotlight to see how the SmartVac project aims to produce novel HBV and HCV antigens with improved immunogenic properties in plants and mammalian cells, following computer modeling and complex molecular and functional analysis for vaccine development. Dr. Nichita, thank you for having us today in your research laboratory. How are you?
Thank you for being here. We are fine, thank you.
How did your passion for biotechnology research start?
I was trained as a protein biochemist, and then I have slowly moved to virology during my postdoctoral studies at the University of Oxford, where I had to study a pestivirus that was then used as a model for hepatitis C virus. And I also had to investigate some drugs, some antiviral drugs that were targeting the viral proteins. And it was then when I understood how beautifully my experience, my knowledge on protein biochemistry could be transferred to viral proteins and virology. And in fact, to come back to your question, what is biotechnology? Biotechnology is just a branch of applied science that deals with organisms, with living organisms, and use them to produce different products that are not normally produced in those cells. For example, proteins that are important for industrial use or for biomedical use, such as hormones like insulin or enzymes or other proteins like antigens. And from that perspective, we are all using biotechnology at some point in our research, because even basic research projects have to find solutions and applications to challenges that our society is facing. So at some point we are all doing some biotechnology research in our projects. And to be honest, this is an age of interdisciplinary science, and we must all embrace related disciplines that are not exactly our comfort zone, and because they might bring different angles to our research, and that's exactly what we are doing in this project.
Speaking about challenges, which are in fact the main causes of B and C hepatitis, and how can vaccine help us?
Together, hepatitis B and C viruses infect more than 500 million people worldwide, and sadly more than 1 million people will die every year of the consequences of these infections. When infection becomes chronic, because this is what the hepatitis B and C viruses do, the disease evolves to fibrosis, cirrhosis, which is repeated cycles of necrosis and liver regeneration, followed by hepatocellular carcinoma, which is the fourth leading cause of cancer deaths in the world. So obviously these are big numbers, and it's a big concern about hepatitis B and C viruses, and it's very important to understand how these viruses are transmitted. There are two main ways of viral transmission. One is the vertical ways, which is from an infected mother to an infant, to her child during birth. And the second way of transmission in adults mainly through open cuts, through blood spots, or through blood transfusions, by sexual transmission, and we must understand that these viruses are very, very infectious, and they can be transmitted even if the blood spot is not visible by eye. And coming back to your question was how a vaccine can help tackle these viruses. So basically there are two important ways that we can use to fight a viral infection. One is prevention, and second is treatment. And there is a common knowledge in medicine, and also a common sense, I would like to say, that prevention is much better than treatment. And this is what applies to these viruses as well. So let's see how things are with hepatitis B and C, because they are a little bit different. With hepatitis B, we have a vaccine that is quite efficient. It gives a protective titer in about 90 to 90% of injected people. But there is a small problem associated with it, and that is, as I said, 10% of people are not responders, and amongst these are people with chronic kidney disease or liver disease or diabetes, or people that are under immunomodulatory treatment. So these are non-responders in general. So we need to do something for them as well. And the second problem is that this vaccine is administered in 3 doses. People don't usually comply with this vaccination scheme and they drop out. For HBV, there is a treatment that can alleviate the symptoms, can reduce the viral load, but cannot cure the disease. So in other words, today we don't have a cure for hepatitis B virus. For hepatitis C, the things are quite different. We do not have a vaccine at all, and that relates to the instability of the RNA genome, because hepatitis C is an RNA genome as opposed to hepatitis B virus, which has a DNA genome and which is much more stable. So we do not have a vaccine, but instead we have a very good treatment. I would say it is called actually a revolutionary treatment because it's in an interferon-free treatment. So it doesn't use interferon anymore as in the old days, but rather a mixture of direct antivirals that act on replication and also on protein processing. And these are very, very efficient and work in more than 90% of treated patients. So we do have a treatment, but we do not have a vaccine. But why is a vaccine so important? And we have this picture from the hepatitis B vaccine. When it was introduced for mass vaccination in 1982, the cases of hepatitis B have dropped significantly, and it is assumed that more than 4.5 million children have been spared from this infection due to the fact that they were vaccinated at birth. So yes, the vaccination is important, and we cannot hope to eradicate these viruses in the absence of a vaccine. And I think these numbers speak for themselves.
Currently, you are involved in a collaborative research project funded under the EAA grants. How did the idea of this project come about?
It actually started from our Norwegian collaborator. Our colleagues in Norway were looking for a partner in Romania for a joint application for this EAA funds that the call had just been launched in Romania. And they are the true biotechnologists in this project. So they were looking for somebody in Romania, for a team who could help them produce some proteins in plants, because they are working with plants, proteins that are important from a biomedical point of view. And they were searching the internet and they found me and my team. And our interest in studying hepatitis B and C viruses, by the way, that's why it's so important to have an up-to-date, friendly, informative website. So they found us and they contacted me. They have contacted me, and they asked me whether I would like to collaborate with them on something that is important for them too. And so we discussed about our interest in hepatitis B and C viruses, and we said, well, the most important thing that we can do by using your expertise, our colleagues' expertise in plants, would be to produce these viral proteins that are exposed on the surface of the virus and that are the main target for the immune response, produce them in plants, because this is a very easy and less expensive way to produce recombinant proteins than in our mammalian cells. So we discussed this idea and we liked it a lot, and we worked on it, and it developed later in a full proposal that won the competition not once but twice, because actually this is the second grant that continues our previous collaboration that had started 6 years ago. So this is how it all started, and because we needed immunology expertise to test our antigens to see if they trigger an immune response and how important this immune response is, we also contacted the Cantacuzino Institute from Bucharest, and they have joined our consortium.
And which are the main hypotheses of your research project?
We have some important questions that we want to answer in the frame of this project. So first is, can we design better hepatitis B and C antigens for the potential development of new vaccines? Is that possible? Because obviously we're not the only ones who investigate this field. This is a very, very hot field. Everybody's trying to find a vaccine against HCV and also to improve the current vaccine against HBV, as I mentioned earlier. So that would be the first question. And we address this question by using a computer modeling approach to design better HCV vaccines, and a rational design approach to construct these HBV antigens that we are going to use to develop these new vaccines. That was the first question. So the second question was, okay, we have these newly designed antigens, but would it be possible to produce them in plants? Because plants and mammalian cells are very different. They are all eukaryotes, but they are different. They have different, at some point, different post-translational mechanisms. So we needed to respond to this question. Is it possible to produce them? If we do produce them, is their quality good enough to make them good antigens to induce a protective immune response? That was the third question. And the fourth question was, would it be possible to use these antigens for an oral vaccine? In other words, could we express them in plants that are edible? That means we can eat them as raw plants, for instance, in lettuce, the common salad, for instance. Would it be possible to produce them in these plants and then use them for an oral administration without having to purify these antigens? In other words, you just eat the salad and you get a boost of your immune response and possibly a protection against this virus. So that was another crucial question we wanted to answer. So this would be the major questions that we wanted to respond in this program.
Are there any partial results that you would like to share with us?
Yes, thank you. We have many interesting results so far. First of all, we managed to produce these proteins in Nichita bentamiana. This is a common tobacco plant, and in lettuce, and also in plants with humanized glycosylation. I know it may sound like science fiction, but indeed today we can produce plants that have humanized glycosylation. They are engineered in such a way that the glycans that are attached to the proteins resembles more the glycans that are found in human cells. So we are now using these plants, and we could express this antigen in these plants, and they resemble indeed very much the antigens that are produced in physiological systems. So that was very good and a result in absolute premiere. Secondly, we have managed to produce the HCV antigens in both Nichita bentamiana and in lettuce. And moreover, we demonstrated that these proteins are of excellent quality. They are basically the same as the ones that the virus produces in our cells. And moreover, we were able to demonstrate that these antigens induce an immune response when they are injected in animals. We use animal studies with the Cantacuzino Institute, as I mentioned earlier, and they use mice to demonstrate this immune response. And we were very happy to understand that these antigens are so powerful that their immune response is actually much higher, much better than the immune response induced by the current hepatitis B vaccine. For example, if we are to compare the hepatitis B antigens that we produced in our project with the hepatitis B antigen that is found in the current vaccine that is on the market. So that was an extraordinary result.
How did the pandemic influence your work so far?
It did influence our work. Unfortunately, at some point during the first lockdown, we had to slow down a little bit our activities. But luckily, we were able to recover these delays. And I have to thank my team and my young PhD students who were very, very helpful and understood that we must work harder to recover these delays. It was a similar situation in Norway. Our colleagues had a more a longer lockdown, and they didn't have access to the greenhouses where the plants grow. So they also had some delays in their activities, but they are slowly recovering these activities. But on a different line, this pandemic, I think, has also helped us understand how important it is to be prepared, to have all the knowledge and all the tools and all the expertise and the professionals that know all this, how to manipulate all these methodologies regarding virus infections, to be able to respond quickly to such a pandemic as the very recent SARS-CoV-2 epidemic or pandemic. Because what we do now in this project can be very easily translated to any other virus in the world. And actually there are groups that now have expressed this very famous spike protein of SARS-CoV-2 in plants already. And then they are using it to develop potential vaccines against SARS-CoV-2. So this is what I'm saying. It is very important to be prepared and to have all the knowledge and to have all the, all these tools and all the funding at some point is very important as well to be able to quickly respond to these emergencies.
Your research is part of a collaborative project which involves an international research team and a partnership with different research organizations from Norway and Romania. What does it mean to you and for the team you are part of such a partnership? And in fact, what are the benefits?
This partnership was extraordinary for each partner of the consortium. And I'd like to answer your question by saying that none of the involved partners could have performed these activities, the activities of these projects alone. Because the Institute of Biochemistry, which is the promoter of the project, has all this expertise in protein biochemistry and virology that is necessary to put up together all these activities. Our colleagues in Norway are the true experts in plant biotechnology, so we couldn't have used plants without them. And also we needed an expert partner with expertise in immunology to actually test our hypothesis and to validate it, and that is the Cantacuzino Institute. So in this way, what we have together is a mixture of expertise and complementary competencies, I could say. And another benefit of this collaboration is that we can circulate among us all the knowledge that we produce in this project, the molecular tools that we produce, and also have this exchange of even culture at some point, because this is being the second project already. Our relationship, I could say, has evolved almost at a personal level, and this is very good because the project is working very well. And it was also very beneficial for our PhD students because we have several involved in this project, because we could send them to Norway, for example, to be trained in plant biotechnology. Not so much at the moment because of the pandemic and the restrictions to travel, but this is in plan, and we will do that in the next future when this pandemic is over, hopefully soon.
What are the next steps of your research?
The next steps would be to validate our results. Obviously, as I mentioned earlier, these are preclinical studies. So what we deliver now is a proof of concept that our product works, is efficient in certain conditions, which is the lab conditions. So, and what we produce is not toxic. It's very important to have this toxicity profile, because a vaccine, okay, it has to be efficient, but at what cost? Because in our preclinical studies, we are using mice. Mice are the preferred animal studies to do this kind of experiments, but we will be using a much better animal model, which is a mice with humanized immune system. How does this work? These mice have a compromised immune system of their own, and they are repopulated with blood cells from healthy human donors. Even next year, starting with next year, we will be testing and validating our results in these new animal models. And again, as I said, we'd like to emphasize more this possibility to use this vaccine as an oral vaccine, in oral administration. We believe this has a great potential, because we would, in this way, reduce the number of injections, if we could prove that administration of a vaccine by oral route can actually boost an immune response.
What do you think will be the social impact of your research?
This is a basic research project, so what we do here is deliver a proof of concept that the antigens that we design in this consortium are working at least in the lab conditions. So obviously we will not have a vaccine tomorrow, that's for sure, so therefore the impact on social life will not be immediate, but there will be an impact certainly, because all the knowledge that we have accumulated in this project will certainly contribute to the development of new vaccine that can be produced in a most cost-efficient way, because we need affordable vaccine. What are affordable vaccines? Cheap vaccines that can be used, for example, in low-income countries for people who need it the most, because it's there where the prevalence of these viruses is highest. So in this sense, we are absolutely convinced that the knowledge that we generate in this project will contribute to save millions of lives, not now, but in the future.
And at the end, if you were to say a few things that people should remember about your project, what would you say?
I would say that when they prepare, for example, a green salad for their lunch or for a dinner, to imagine that not in a very long time, hopefully, by eating a salad, this will provide not only the necessary supply of nutrients and vitamins and fiber that are very useful for our health, but also if this salad contains viral antigens, this may boost our immune system and may protect us from infection. And this is not science fiction at all, it's just science.
Thank you for watching Research in the Spotlight, a podcast for scientists and passionate science people made under the EEA Grants Research Program by the Executive Agency for Higher Education, Research, Development and Innovation Funding as program operator. Until the next time, like and share this episode if you think science is our key to a better future.